Your teacher today is Dr. Bbosa Science. Dr. Bbosa is a teacher at the Science Foundation College, the best school that teaches sciences. Today we are going to revise structured equations of photosynthesis. Question 1. Figure 1 shows the concentration of glycerate phosphate GP and ribulose biphosphate RIU BP during an investigation in which a sample of Chloroplast was allowed to photosynthesize at very low and very high concentration of carbon dioxide. The variation of GP and ribulose biphosphate are shown in the graph. A. Explain the change in the concentration of ribulose biphosphate at high carbon dioxide levels. At high carbon dioxide level, the concentration of ribulose biphosphate is low because it combines with carbon dioxide and water to form glycerate-free phosphate. You remember in the Chloroplast cycle, ribulose biphosphate combines with carbon dioxide and water to form glycerate-free phosphate. That's why the concentration of glycerate-free phosphate in the graph high, while the concentration of ribulose biphosphate is low. 2. At very low carbon dioxide concentration. At very low carbon dioxide concentration, ribulose biphosphate concentration is high because it is being regenerated from the triose sugar and is not being used to combine with carbon dioxide. B. Suggest why the concentration of GP falls when the level of carbon dioxide is reduced. Because there is not enough carbon dioxide to combine with ribulose biphosphate to form GP. In the Chloroplast cycle, ribulose biphosphate combines with carbon dioxide to form GP. C. Name 2 factors that must be kept constant in the investigation. That is temperature and light intensity. Because chlorophyll is already constant in the chlorella. D. Give 4 differences between cyclic and non-cyclic photophosphorylation. Differences between the cyclic and non-cyclic photophosphorylation. Electron transport is cyclic in cyclic photophosphorylation. In non-cyclic photophosphorylation, electron transport is non-cyclic. 2. In cyclic photophosphorylation, only photosystem 1 is involved. In non-cyclic photophosphorylation, photosystem 1 and 2 are involved. In cyclic photophosphorylation, produces ATP only. In non-cyclic photophosphorylation, produce ATP and NADPH. In cyclic photophosphorylation, photolysis of water does not occur. In non-cyclic photophosphorylation, photolysis of water occurs. Number 2A, what is chemosynthesis? Chemosynthesis is the process by which organic compounds are synthesized from inorganic raw materials using energy from oxidation of inorganic compounds. B. Outline 3 ways in which photosynthesis in a purple sulfur bacteria differ from that of higher plants. Differences between purple sulfur bacteria and high plants. We have bacteria and high plants. In the bacteria, the source of hydrogen is hydrogen sulfide for reduction of carbon dioxide, while in the plant, the source of hydrogen for reduction of carbon dioxide is water. Sulfur bacteria produce solid sulfur as a byproduct. Higher plant produce gaseous oxygen as a byproduct. Sulfur bacteria use bacterial chlorophyll, while higher plants use chlorophyll. C. Explain why it is possible for photosynthetic and chemosynthetic bacteria to coexist in oxygen-free environment. The photosynthetic bacteria release oxygen used in respiration by both photosynthetic and chemosynthetic bacteria. It also releases organic compounds which can be used by respiration by chemosynthetic bacteria. Chemosynthetic bacteria will produce hydrogen sulfide and other chemicals that are used by photosynthetic bacteria. Part D. State the importance of chemosynthetic bacteria in nature. 1. Chemosynthetic bacteria decompose and recycle matter. 2. They synthesize organic materials used by other organisms. 3. The figure below shows light saturation curves of photosynthesis for plants of the same species growing under different light intensities. The graph is as below. A. Compare the effect of intensity on carbon dioxide uptake in the two plants. The question requires that we state the similarities and then the differences. First, similarities. The rate of carbon dioxide uptake increases initially at low light intensities and then decrease gradually at higher light intensities to reach a constant value. So each graph levels off at high light intensity. Differences: light plant, shade plant. Initial rate of carbon dioxide uptake is low at low light intensity. Initial rate of carbon dioxide uptake is high at low light intensity. In the light plant, carbon dioxide uptake starts below zero light intensity, while in the shade plant, carbon dioxide uptake starts at zero light intensity. Light plant achieves higher saturation value, while shade plant achieves lower saturation value. B. Describe the state of light plant at point P. The plant is at a state of compensation at point P. There is no net uptake of carbon dioxide because the rate of photosynthesis equals the rate of respiration. C. In region Y, the biomass decreases because the rate of respiration is higher than the rate of photosynthesis. D. State an environmental factor that affects the shape of the graph other than carbon dioxide. That is temperature. 4a. How does the synthesis of organic compounds in photosynthetic bacteria differ from that of green plants? So green plants obtain hydrogen for reduction of carbon dioxide from water, while photosynthetic bacteria obtain hydrogen for reduction of carbon dioxide from hydrogen sulfide. Green plants use chlorophyll, photosynthetic bacteria use bacterial chlorophyll. 2. Chemosynthetic bacteria. Photosynthetic bacteria obtain energy for synthesis of organic matter from sunlight, while chemosynthetic bacteria obtain energy from oxidation of inorganic material, such as hydrogen sulfide and ammonia. 4b, the figure below shows a scheme of reaction occurring in a chloroplast. Light stage, ATP, NADP. B, NADPH, NADP. Then Calvin cycle. W is taken in, X is given out, Y is taken in Calvin cycle, Z is given out. 1. Name the chemicals represented by the letter W. W is water, X is oxygen. Calvin cycle takes in carbon dioxide and releases sugars. Triose sugar. 4b2 indicate by means of arrows on the diagram the direction of transfer of substances occurring in cycle A and B. So light stage produces ATP, which is used in Calvin cycle. Also light stage produces NADPH, which is used in Calvin cycle. C. State-free adaptations of chloroplast for photosynthesis. 1. It contains chlorophyll for trapping sunlight. 2. Has extensive membrane structures for increasing surface area for attachment of enzyme. 3. Has necessary enzymes for photosynthesis. 4. Has a large surface area to volume ratio to increase the rate of diffusion of carbon dioxide. 5. It is thin for light penetration. If you wanted more, it contains DNA for fast multiplication. And 7. It contains ribosome for synthesis of enzymes. 5a, giving examples, differentiate between photosynthetic bacteria and chemosynthetic bacteria. Photosynthetic bacteria manufacture carbohydrates from carbon dioxide and hydrogen sulfide using energy from light. Carbon dioxide plus hydrogen sulfide, you get carbohydrate plus sulfur. Chemosynthetic bacteria manufacture carbohydrates from water and carbon dioxide using energy from oxidation of inorganic chemicals such as iron 2 salts and ammonia. B. Explain how certain bacteria which require light for photosynthesis survive under weeds in the ponds and rocks. Bacterial chlorophyll absorb light at different wavelengths from that absorbed by plant chlorophyll. Thus, bacteria find useful spectrum after plant light absorption. Secondly, the rock supply hydrogen sulfide, which provide hydrogen for reduction of carbon dioxide in photosynthesizing bacteria. 6a, what is photophosphorylation? Photophosphorylation is the formation of ATP from ADP and inorganic phosphate using sunlight as the source of energy. B. Where in a plant does cyclic photophosphorylation occurs? It occurs in the thylakoid membrane of the chloroplasts. C. Describe the process of cyclic photophosphorylation. Light strikes chlorophyll, photosystem 1. An electron is excited and taken up by an electron acceptor. An electron acceptor hands the electron to PS1 through electron carrier proteins. In the process of electron transfer, hydrogen ions builds up in the cyclic space, and the passage of hydrogen ions out of the cyclic space provide energy for synthesis of ATP. D. What's the importance of cyclic photophosphorylation in photosynthesis? It synthesizes ATP used in the carbon cycle for reduction of carbon dioxide to produce carbohydrates. Number 7A, name the organelle most important in photosynthesis and respiration. Photosynthesis is chloroplast. Chloroplast. Respiration is mitochondria. B. Give 4 similarities and 5 differences between the two organelles. Similarities between mitochondria and chloroplast: both have structural DNA. 2. Both are bound by a double membrane. 3. Both have matrix. 4. Both produce ATP. 5. Both have extensive membrane system. And 6. Both reproduce by cell division. Differences: chloroplast contain chlorophyll, mitochondria lack chlorophyll. Chloroplast synthesizes sugars, mitochondria break down sugars. Infolding in chloroplast is granular, in the mitochondria is crystal. Chloroplasts are green, mitochondria are colorless. Chloroplasts occur only in plants. Mitochondria occur both in plants and animals. Thank you so much for listening to this video. These are just samples of structured questions. Find more resources of biology on digitalteachers.co.uk. On the top bar menu, you will see A level. Then when you click A level, you will get a drop-down menu which shows biology. When you click biology, it will give you another drop-down menu which shows A level biology. Then when you click A level biology, you will be able to get more resources in the biology. I'm requesting you to subscribe to promote my videos. Note that subscription for these videos is entirely free. You just click the subscription button, you fill in the required information, and that will be done. If you have enjoyed the video, share it with your friend. Next will be essay questions in photosynthesis.